When the sound has travelled down the meatus, its energy causes the eardrum or tympanium, which forms the end of the auditory canal, to vibrate like a speaker diaphragm. Although the eardrum is regarded as part of the outer ear, it is physically joined to the structures, the ossicles, of the middle ear, and together they transfer the vibration of the eardrum across the middle ear to the inner ear. The middle ear is filled with air, like the auditory canal, but its connection to its air source is the eustachian tube, which exits in the back of the throat. The pressure either side of the eardrum can therefore be equalised, even though the source of air is different either side of the eardrum. The eustachian tube is normally closed, but it opens about every 3 minutes when one swallows. The eustachian tube is designed as an outflow valve to drain unwanted substances, and to release or increase pressure in the middle ear when pressure becomes higher or lower than the ambient pressure. For example, during an ascent, when the pressure in the auditory canal reduces quicker than in the middle ear, the eustachian tube opens to let out some of the middle ear pressure. In the descent, air pressure in the auditory canal increases quicker than in the middle ear. In order to equalise the pressures either side of the eardrum, the eustachian tube has to let air into the middle ear. It may be necessary to force air into the middle ear by pinching one's nose and blowing, known as the Valsalva procedure, or by swallowing. For this reason, a person is more likely to experience pain in the ear, otic barotrauma, during the descent. Any blockage of the eustachian tube, perhaps because of a cold, can cause pain in the middle ear due to the pressure imbalance. Children are more prone to this, as their eustachian tubes are not fully developed. The problem which the middle ear has to overcome is how to transfer sound received through air as far as the eardrum into sound in the inner ear, which is filled with fluid, without a huge loss of power. Impedance means the resistance of a system to an energy source. There is a huge difference in terms of impedance to the passage of sound waves between air and water. The specific acoustic resistance of air is approximately 42 ohms, while that of a small body of water is about 9,000 ohms, a loss of about 30 decibels. If impedance did not match, most of the sound would be reflected at the air-fluid interface, and would be unable to generate a pressure wave within the fluid. Just such an interface exists between the middle ear, which is full of air, and the inner ear, which is full of fluid. There are 3 bones in the middle ear, collectively known as the ossicles. They are the malleus, the incus, and the stapes. In lay terms, these are also known as the hammer, the anvil, and the stirrup. They are all hinged together and form a bridge between the eardrum and the oval window on the cochlea, which is part of the inner ear. As we have just seen, there is a loss of about 30 decibels of volume when sound travelling through air tries to travel through water. The ossicles solve the problem of this loss of volume in 2 ways. Firstly, they provide a leverage ratio of 3:1, resulting in greater pressure on the oval window, giving a gain of about 2.5 decibels. Of greater effect is the area ratio of the eardrum to the stapes. The effective area of the eardrum is about 47 square millimetres. The area of the base of the stapes, where it connects to the oval window of the cochlea, is about 3.2 square millimetres, giving an area ratio of the 2 surfaces of 14.7:1 and a decibel gain of 23. Remember that pressure is equal to force divided by area. These three little bones have therefore recovered the decibel loss between air and water caused by their impedance mismatch, and they have caused sound waves created and transmitted in air to have the same effect in a fluid. The outer and middle ear deliver sound to the inner ear at a suitable pressure and amplitude for the sound to be converted into a neural message to the brain, which then interprets the message as the original sound. The inner ear is located in line with the outer and middle ear, about 5 centimetres into the skull from the pinna. There is a cavity, called the bony labyrinth, within the temporal bone, which is filled with a liquid called perilymph. Suspended within this cavity is a similarly shaped membrane, creating a second chamber, the membranous labyrinth, filled with a fluid called endolymph. The whole structure is called the inner ear. One half of this structure deals with balance and is called the vestibular labyrinth or vestibular apparatus, and comprises two parts, the vestibule and the semicircular canals. We will look at this later. The other half, called the cochlea, deals with sound. The cochlea is wound up like a snail in order to take up as little space as possible. If we unwind the cochlea, it is easier to understand its anatomy. The cochlea encloses the scala media, filled with endolymph, while the bony labyrinth which surrounds it is in two parts, called the scala vestibuli and scala tympani. At one end of the scala vestibuli is the oval window, which receives the pressure variations from the stapes. These pressure changes are partly absorbed by the scala media, while the remainder continues round to the scala tympani and back through the round window to the middle ear, thereby returning the excess pressure to normal before the next stimulus arrives. This wave of pressure caused by the stapes at the oval window causes a complementary movement of the fluid in the scala media, which contains the mechanism for converting this pressure wave into an electrical message to the brain. Let us look at the scala media in a little more detail, as this houses the mechanism for converting the sound pressure wave into an electrical signal to the brain. Separating the scala media and the scala vestibuli is a membrane called Reissner's membrane. Separating the scala media from the scala tympani is the basilar membrane. Pressure waves entering the oval window move along the scala vestibuli, which causes similar movements in the Reissner's membrane and the basilar membrane. The basilar membrane is internally formed by thin elastic fibres, tensed across the cochlear duct. Nearest the oval window, the fibres are short and thin, but they get progressively longer and thicker further along the membrane. Because the fibres are under tension, they produce an effect like guitar strings, in that the fine strings make a high-frequency noise, whereas the thick strings make a low-frequency noise. Similarly, the thin fibres at the oval window end resonate to high-frequency sounds, up to about 16,000 Hz, while the thick fibres at the apex end resonate at the lower frequency sounds, down to about 40 Hz. Try this interactive demonstration by placing the mouse over the frequency displays. The basilar membrane therefore acts as a noise spectrum analyzer, with the various frequencies of the wave resonating at different places along its length. How, then, is this movement of the basilar membrane communicated to the brain? Along the length of the membrane is a sense organ, called the organ of Corti, which contains 4 rows of hair cells, a single row and 3 rows. These hairs are rooted to the basilar membrane, where they are connected to the ganglions that connect to the brain. Their tops lean against the tectorial membrane, which is part of the organ of Corti. When the basilar membrane moves as a result of a pressure change in the endolymph, the hair cells move forwards and backwards against the tectorial membrane. The single hairs tilt with the movement of the membrane, and this causes a tiny voltage pulse, called an action potential, to be sent down its afferent nerve fibre to the brain for interpretation. The outer 3 rows of hairs act as amplifiers of the wave. When they bend, they also contract, then bounce back against the tectorial membrane, amplifying the vibration of the basilar membrane so that one can hear very quiet sounds. The electrical charge occurs as the hairs tilt, allowing positive potassium ions from the endolymph to enter the hair, which has a lower potassium charge and therefore creates a large positive potential difference. The process is reversed as the hairs return to their former position. This change in polarization is detected by the nerve connected to each hair and relayed to the brain. The pitch is relayed according to the position of the hair cell along the basilar membrane, while the volume is signalled by the discharge rate and number of active afferent nerve fibres.